Our nanoneedles are like 1,000th of a human hair. What you can do with nanoneedles is get the same information that you would get from the tissue, but without taking the tissue away.
Welcome to Health in Hyperdrive, the podcast where we explore the breakthroughs shaping the future of healthcare. I'm Dominic James, and today I'm joined by Dr Ciro Chiappini, a reader at King's College and a pioneer in nanomaterials with 5 international patents to his name. In the Chiappini Lab, Ciro and his team create smart materials that can detect, study, and even control biological processes at the molecular level. Ciro, welcome to Health in Hyperdrive.
Well, thanks for having me, Dom.
So, Ciro, your work sounds like science fiction, I would say. You put tiny needles that work inside the body at the molecular level. What is that all about? It sounds crazy.
Yeah, I mean, okay, think about a bed of nail, okay, but just extremely small. And instead of yourself sitting on it or fuck here, the cells in your body sit on it. And when they sit on it, they really like to attach there and to spread. And that allows the nanoneedles to poke into the cell. And when they poke into the cell, they can either deliver a drug or they can extract the content of the cell for us to analyze it later. Doing these, we can do things such as gene therapy by delivering genes into the cells, or molecular diagnostic by picking out RNA or proteins to look at what's in there.
And can you talk a little bit about kind of gene therapy? Like, what is it that these tiny needles are delivering to the cells?
Yeah. So gene therapy occurs when either someone has a genetic defect that needs to be corrected, or, for example, if in aging, some cellular processes stop working. And what you do is that you deliver either copies of functional genes to the cells, or you go there and you correct the existing genes to basically get the correct form of it.
And then for you didn't call it precision medicine, but you said taking things out or monitoring a cell. Like what is that? How does that work?
So the way in which we call it molecular diagnostics, and that is what supports precision medicine. So it allows you to tell at the molecular level how a disease looks like, right? Diseases such as cancer, they may all have the same symptoms for a person, but these symptoms can be caused by very different processes occurring within the cells. And what we can tell is allowed to distinguish between a type of cancer that is caused by certain protein, for example, I don't know, in breast cancer is famous HER2, or other type of cancers that are caused by different molecular processes, for example, triple negative breast cancers. And these means that you need very different treatments for each type.
And typically in cancer treatment, you have like biopsies, and so I guess that's fairly invasive. You'll have a biopsy, and they'll take a lump of your tumor out and do the analysis and make some decisions. What's the difference between, say, that approach and what the nanoneedles that you're building can enable?
Yes. So exactly as you said, when you do a biopsy is an extremely invasive process, and you cannot always do it because you may be taking away a piece of tissue that cannot be taken away. For example, in the brain, this happens very often. I mean, you have functional parts that you cannot remove, and if you want to understand what's happening there, you're out of luck. Or in some cases, you may want to monitor how things progress over time, and you can't really have patients come and take a biopsy every 3 months or every 6 months. I mean, even taking a biopsy every year is something that is quite painful and would put many people off doing it. What you can do with nanoneedles is get the same information that you would get from the tissue, but without taking the tissue away. So you just apply the needles to the piece of tissue that you want to analyze, and instead of analyzing the tissue itself, you do the same type of analysis on your sort of impression of the tissue, on your imprint of the tissue that you get on the needles.
All right. I think maybe now's a good point to talk a little bit about your background. Like, how do you, like, you have a engineering background, I think. Like, how did that even start? How do you get into nanotechnology?
I think even sort of further than that, my sort of undergraduate is in solid state physics. And I did as a thesis. So in Italy, we have sort of an kind of an MSci type of approach where you get all the way to a master thesis. And in my master thesis, I was actually working on gas sensors from nanostructured materials. And because I specifically told my advisor that I didn't want anything to do with cells or anything living, because those things are unpredictable. You don't know what they do. But as I was doing my thesis, the sort of the person next to me was working on using these nanostructural surfaces for growing cells. And in the end, I found out that that looked a lot more exciting than my very boring gas sensing project.
And what was kind of exciting about that?
Well, it was, I think, really the unpredictability, the fact that you would just put your cells on and then look at what happened and try to understand and characterize and sort of look into this very complex system, while the things that we were doing were comparably a lot simpler and dull. And so I got really excited about, doing materials and nanomaterials, especially for biology. And back then, it was the time where there was a huge boom in nano delivery systems. Lipid nanoparticles were just coming to the front, and there were a lot of people had just started to explore gold nanospheres and this kind of thing. So when it was time to decide my PhD, I went to my advisor and I said, well, actually, I changed my mind. I really want to do something that has to do with biology. What's interesting out there? And I think one great advice that he gave me is, well, you could learn how to characterize something, but it's going to be a lot better if you learn how to make something. And so coming from, again, a materials science background, what I decided to do for my PhD was fundamentally micro and nano fabrication for biology. And back then I was making sort of micro and nanoparticles from this material, which is porous silicon, using the semiconductor industry processes. So the things that you use to make your computers. And so I got a lot of cleaner experience, and I started to work at the interface between materials and cells. And then progressively from there, I moved more and more into the biology. During my sort of postdoc, I worked a lot at the sort of cell material interface, doing all of these delivery studies and sensing studies. And then as I become an academic, I'm now in a regenerative medicine and cell biology department. So I think I've moved from the hard physical sciences all the way into the biological sciences.
Yeah, that's quite a journey. What's motivating you?
I love learning. I think that's the most interesting thing, and I always captivated by what is new, and especially what is new to me. Right, so uh, lear- progressively learning about, for example, molecular networks and gene regulatory networks, and then cancer biology, which is something that I didn't learn until my PhD. And then later on, stem cell biology and reprogramming and all these kind of very interesting things. They were extremely exciting. And I mean, I don't know, I'm a hoarder for knowledge. And that sort of maybe in a way it comes across also in the type of research that we do. I do kind of a bit of left field research in the sense that I, I do things that other people, I don't know whether they don't like to do or they're not interested, but they don't really fall into a mainstream, because if there's already a lot of knowledge about that, it maybe doesn't excite me as much.
So is it that you're kind of very curiosity driven? If I take my perspective, and I feel like, and I think this is true for everyone, you can apply your skill sets in many different ways, and I'm curious like you about many different things. But for me, I've chosen healthcare because that really motivates me, and I really want to build companies that will make an impact and create better outcomes for people. So for me, the mission is my motivation. for you, is it the mission, or is it the kind of the technology and the creation?
I think that for me, the mission is the creation. I mean, I really, really, really like to explore new frontiers. And obviously, uh, it's fantastic that the things that we do benefit patients and society at large. And it is extremely important. And I think that it's an exciting um way to explore these aspects. I want to make something new, but I also want to make something that is relevant and that can help uh, people.
And can you tell me a bit about the Chiappini Lab? I guess that's named after you. It didn't work the other way around. They didn't have a lab and
Yeah.
found a guy called Chiappini.
No, I mean, well, it's also sort of, we call it bio-nano engineering lab as well. So sort of, labs in academia are often called after the PI because it's sort of recognizable that way. But things that we're interested in are, on the one side, extremely translational, as we were saying, delivering genes better and doing precision medicine. And then on the other side, we also like to explore fundamental science and cell material interfaces largely. So understanding how our cells respond to the presence of materials, especially nanostructures, and how the interaction works and because this is extremely important, is fundamental to making nanomaterials work for healthcare.
And what would you say are the major sort of breakthroughs or pivot points that you've had in your kind of recent career?
Well, I think the two biggest things that we've done recently, one is that we've been able to efficiently deliver CRISPR vectors, so do gene editing in primary human cells. And we are in the process of doing that in animals, and that is extremely challenging. And the second one is this recent work that we've done in precision medicine, exactly where we can do these sort of, longitudinal or, monitoring over time of precancerous or cancerous lesions uh, without taking out the tissue in a non-invasive way. These are the two big breakthroughs.
It's really incredible. Do you mind like explaining a little bit CRISPR for just for the audience out there and maybe me?
Yeah, no, I think to me the simplest analogy is with film editing, right? I mean, when you shoot a film, you shoot the entire scene, uh, and then you can consider that as your gene. But then in there may be part of it that are wrong, that don't work. But after the fact, you can edit them out. And CRISPR can do exactly that in a few different ways, exactly in the same way as you do it in cinema. You can either take out the bits that don't work and then put together the parts that work. Okay. You can insert an additional bit in between wherever you want. Okay. Or you can change a part. Okay. And these are fundamentally the three things that you can do, but on genes.
And so you do that in genes, and then the technology that you have is you're able to inject these genes into cells.
It is a bit different in the sense that the big challenge, so the way in which you do it is you want to do it in the genes that are already in the cell. That is the challenge, right? You want to do it in the patient so that you don't have to introduce external cells or you don't have to introduce anything else. You can correct within the patient cell itself. And the problem is that obviously our cells and our body has developed exactly to prevent these things from happening, because that's what viruses and other things that would harm us want to do. So getting the CRISPR components, the sort of the tools that you need inside the cells is extremely challenging. And what we do is that we build the vector, so the sort of the delivery system, the Deliveroo, that brings the CRISPR to the cells.
The Deliveroo, I love that. Maybe this is a good time to talk a little bit about like, we've talked a little bit about nanotechnology, and I'm saying it like I know what it is. I know that nano is small, and this is like super small technology, and obviously we're talking about working at a molecular level, so it's really tiny. But like, how tiny are these needles? Like, can you give like a reference? I guess like the small thing I'm thinking is the human hair, but
it is extremely challenging because even with the human hair, you have to think that our nanoneedles are like 1,000th of a human hair.
wow.
So I mean, it's really
Well, I mean that's perfect. 1,000th of a human hair. I mean that's super tiny. So, and then how do you build something that small?
as I was saying earlier, we just use the semiconductor industry processes. So the same tools that you use to make your computer chips, right? They're made out of silicon, and it's the same material that we use to make our needles. And you use this combination of sort of patterning and etching away the material or building on that. But it's extremely controlled, and as you can see or you can hear probably from the news, you can now control down to 4 nanometer diameter.
Sorry, how big is a nanometer?
A nanometer is 10 to the minus 9 meters, so it's 1 billionth of a meter.
1 billionth of a meter. That's like, yeah, okay.
Yeah.
You can't imagine, right?
I think that more than the scale, the important aspect of nanotechnology is that materials behave differently when you shrink them to that size. Okay, so the reason why nanotechnology was born is that still when you're at the micron scale, your material largely behaves the same way as these large materials that you see. But when you move down to the nanoscale, you get these so-called emergent properties. So materials gain new properties just because of their size. One example is, people maybe now know about quantum dots, which have gotten into TVs. So your recent TV screen from Samsung are made of quantum dots. These are very tiny nanoscale dots of semiconductors that are fluorescent. The material itself, when it's sort of bulk material, is not fluorescent at all. But when you shrink it down to the nanoscale, then it becomes fluorescent. And other examples are gold, that is yellow at when you see it. When you shrink it down to the nanoscale, it switches its color between red and blue, depending on size. And you can use this information as a sensor, okay? Because if you grow larger particles, they become blue, and if they are smaller, they're red. And you can use combination of molecular systems to grow them of different sizes and tell you what they are.
That's nuts. Gold is blue, and well, okay, I'm gonna stop there. That's just blowing my mind. So maybe as you talked about kind of chip manufacturer processes effectively designing these kind of nano needles. I don't really know how that how chips are really made, but is there a way you could kind of maybe explain in sort of layman's terms? Do you have like a super like blown up picture of what you're trying to build, or it doesn't work on a visual level like that?
Well, I think like every kind of building, it's building, right? So there are three things that you can do. You can make patterns, okay? So you can draw where you want your things to be, and then you can either take away material to leave the pieces that you want, or put material on. And you do that in combination, and you can combine layers of different materials one on top of the other, and that's how you make them. But then the way in which you do it is extremely complex. It requires machinery that allows you to do it at very small scales.
But the design process, is that in the same way you would design like an engineering part, like a CAD design?
Yes.
It's exactly like that. It's just when it gets created, it's a microscopic scale.
The design process is exactly the same, and that is what the semiconductor industry uses as well. Your chips are designed with what they call vias, and they're effectively lanes in which the current moves around, and they draw them on these CAD maps. And then obviously each layer has to be made of a different material and so on and so forth for these devices to actually work. But then they're printed onto these silicon substrates. And You go from engineering design to manufacturing, and then the final stage is packaging. So these are all sort of nanostructured and microstructured objects. You then have to bring them back to the macroscopic world and interface them with the PCBs or a circuit board, and then eventually with your plug and mains.
Can we talk maybe a little bit about the kind of the process of that? Um, so I'm going to ask a.
Mhm.
It's going to sound naive, like dumb question. So you've got tiny needles, and in your case, it's tens of millions of them. It's not like one like little syringe or something like that. And it's manufactured out of silicon, but this thing is still like super small. How do you go from this tiny thing to kind of attaching that to the cell or even getting it into the body? Like, how does that like
So
kind of all work?
in the simplest form, so you make these objects onto what is called a silicon wafer, which is a disc that can be anything between 10 to 30 centimeters, which is the standard in the industry. And you make we can cover all of it with needles, okay, and then you can dice it or cut it up into little squares. And these squares are the ones that we normally use for our basic testings. You just put your cells on top, the cells will grow on them, and that's what we do. You can place them onto tissues, that's equally well done. But the very important thing that we've uh, learned how to do, and that one of the things that we've patented, is that we know how to put these needles onto fundamentally any medical device. We can put them onto bandages for skin wounds. We can put them on contact lenses. We can put them on catheters, balloons, stents, you name it. So we can use the needles to reach any part of the body that is already accessed with the conventional tools that are available in the clinic.
Oh, so that's super fascinating. So that's like effectively you're enhancing just any type of. I mean, you've just said it. I'm just repeating it now. But you're enhancing any type of medical device and bringing that capability to add and remove at a molecular level to any part of the body.
Yes, fundamentally, yes. In fact, one of the first applications that we're thinking of is delivering gene therapy to skin wounds using our nanoneedle bandages. Um, Other applications that we can foresee is, for example, sampling the molecular characteristic of atherosclerotic plaques so that they can help you understand which type of treatment would work better for you. And, I mean, then the sky is the limit.
And can we jump into that example a little bit? Like the bandages one, that's for a layman like me and maybe for others as well, that's a very easy example to sort of comprehend. Um, the needles are super small and they're very precise. How do you target them at the right places if they're just applied with a bandage?
So the thing is, the uh, case on which we're working, or the disease in which we're working with great support for many years from this charity, which is called Cure EB, is epidermolysis bullosa. This is a skin disease that occurs in children, so it starts at birth. And it's a real, I mean, it's a significant tragedy for these families, because it's again, is a defect in a gene that is called collagen 7. And what this gene does is that it links the top layer of your skin to the lower layers of your skin. And when this gene is defective, then the top layer of your skin becomes very fragile and is very easy to be, removed and disrupted. So during their daily life, playing with their friends, going to school, and in their daily activities, these kids lose the top layer of the skin, and they get these like wounds that then can't heal anymore. Okay. And there's no treatment for that. So what we're trying to do is we're trying to deliver the defective gene in the wound. And the advantage of our approach is really that with the needles, we can cover the entire wound and deliver to all the cells that are present in the wound without affecting other cells that are elsewhere that could have a negative or side effect if they were to be delivered this gene.
That's a really emotive. Obviously anything to do with children is massively emotive, and as a father,
Yeah, no,
if you that feels really
no,
personal, and it's really powerful that you're doing
and
that.
it is really, sort of rewarding for us doing this, especially when we consider that, I mean, this is a fairly rare disease, not extremely rare, but there are like 5,000 kids
Mm,
in the UK with the disease. And at the moment, there is no treatment for them. The only solution is palliative care. And this requires bandaging, extensive bandages, which, first of all, impairs their ability to live well, but also take up a lot of time, up to 2 hours every day for the whole family, for the kids that have to wait there. And if we can improve that by, reducing the amount of bandages and reducing the time that they need to be
mhm.
bandaged, and improving their overall quality of life, I think that alone will be an enormous achievement for us.
Yeah, that's incredible. And where is that in terms of, is it in the real world yet, or it's kind of in the lab, or your experimentation, or a testing stage?
I think we're getting out of the lab and into the real world. So we have tested it all the way up to the mouse,
Hmm.
and it does work. So that is very comforting. And but this was always, everything was done with laboratory prototypes. So at the moment we are developing it into a actual medical device, and then we're going to be testing that medical device first for its safety and then for its effectiveness
And
in humans. So we're kind of at that
at that stage.
cusp and transition point.
And what's it's a hard question to answer, but if this thing is, if this example is successful, would you envisage, say, the time frame with which it would become maybe an approved treatment?
We think that it can realistically get into the healthcare system within 5 years, because we can get into men in 2 to 3 years. And then, if things are successful, there can be a clinical trial and an adoption.
And is this technology different from kind of other types of products? I mean, does it require a new way of practicing medicine? Or I guess for this example, almost like maybe the beauty of it is it's not a new way, it's using current practices, just taking it to the next level.
Yeah, I think one of the big advantage of the way in which we're designing our devices is that we want to uh, reduce the amount of change that you need to have from the current practice, right? So we put the needles on the same bandages that are already being used for the therapy. They would be applied in the same way as they're currently applied um, in the when they go to the clinic to get treatment. So we are fundamentally having zero impact on what is the workflow and the practice in the hospital and for the clinical team.
That's incredibly important for innovation. One of the, well, I mean, this obviously is why you're doing it, but like the one of the major barriers, in fact, the major barrier for innovation to be kind of successfully deployed and scale is the workflow challenge. Like changing a workflow is actually really, really hard. So it's really incredible that you're sort of taking that approach, and it's just almost like a copy and paste, put it in, right?
Yeah, no, I mean, it is, because obviously if you change practice, you have to train all the practitioners, right, for that, and you have to make sure that they're proficient and everything. So I think it is a key barrier to innovation. The other advantage is that, being these sort of advanced type of therapies, uh, they would be delivered by specialized units, right? So, for example, in the case of epidermolysis bullosa, there are 6 units in the NHS in the country. So the and they are already very aware of the context and very up to speed with what is the edge of technology. So the ability of working with highly specialized unit also allows to translate this faster than if it were to be delivered in primary care.
Yeah, and I suppose that's also that's a bit the power with rare diseases in a way. Like in rare diseases, they're massively under resourced typically because they're not the things that the big commercial players kind of focus on. So there's a lot of need there, but then I think, as you've described, they're so specialized and discrete. It's quite easy to um, get a grip of the people involved in them and bring people together and actually make a change because it's kind of small enough and manageable enough.
Yes, I see them as being a very good entry point for new technologies for many reasons. Also, the orphan disease designation that is available in Europe and in the US allows to accelerate some of the sort of translational and regulatory aspects of different technologies. And yes, as you said, you have this ability to be extremely focused, both in terms of the partners that you have to discuss with, uh, and in terms of when it comes the time to deploy it. The other big advantage that I see is that in many cases, many of these rare diseases are good model diseases. So epidermolysis bullosa is a good model disease for genetic mutations because it's very well known and very well defined where the mutation happens. The effect is very clear, and so on and so forth. So that allows you to test and validate all the scientific aspects in a very neat and clear way, which, for example, in multifactorial diseases or other diseases that have more variability will be a lot harder to do.
We talked. So this kind of topic is really around gene therapy, and then of course the other dimension you talked about is monitoring disease in a continuous basis. Could you talk about that a little bit more? Like what's the kind of the example there, and then how does that work, and how does that
So
change things?
in that space, we are a bit earlier in the development pipeline, so we're still considering many disease cases. One of which, for example, is monitoring progressively the lesions that you can get in your mouth. And these are typically, lesions that appear as white or red, but they're not necessarily a cause for concern. They can stay in that form for a very long time or forever and not cause and not develop any further. But they can also be precancerous lesions. They can also develop, and sometimes reasonably quickly, into cancer. And at the moment, the way in which this is monitored is by getting into the clinic every 6 months or so. And the clinician, who is an expert in the field, will look at it and, based on their hunch, will say, this looks a bit suspicious, or, oh no, this is fine. And if they say, this looks a bit suspicious, then you have to get a biopsy. And you also have to decide where within the lesion you have to get a biopsy. And it may not be that everywhere is the same. And then again, you can only do this biopsy once in a while. You can't get people back every 6 months and keep biopsying them for many reasons. One of which is that they're not going to come back the second time or the third time. And the second is that the more biopsy you take, the more likely you are to activate all of these processes that actually can help this lesion become more malignant. So the idea is that instead of having to rely on a very educated guess from the clinician, you can, every time the patient comes in, take a sample with your needles and analyze it. And then the clinician has a lot more information and can, instead of making an educated guess, make a precise sort of molecular assessment of, oh, this lesion has not changed from last time. It's still the same. Nothing to worry about. Or, oh, look, these genes have now come up, and this is an indication that these cells have become more motile, or this has become more malignant. We should intervene, or we should monitor it more closely. You should come back more often. We think that this can make a significant difference in both preventative medicine, which is extremely important, and then also in deciding what type of treatment you can get.
And the example you gave was something within the mouth, which I'm assuming that's the place to start because it's kind of accessible. But in theory, you mentioned this before, it could be like, I don't know, for example, a brain tumor that you could be effectively monitoring without having to remove
Mhm.
a part of the brain, which could potentially
So
be.
for the brain, I think that the more compelling aspect is that um, you don't really want to remove bits of brain if you don't have to. And in cancer surgery, uh, they have to make a lot of these very difficult decisions many times, even within the same surgery. And normally, the way in which they make this decision is that they take out a little bit of tumor and then they send it out for histopathology straight away. And what you can do straight away in terms of histopathology is very limited. You get this so called frozen section that, first of all, take a long time to come back. So the surgeon has to wait there for like 20 minutes, half an hour, mm, maybe even an hour until the information comes back. And then it tells them, oh, well, yes, this part is malignant, or no, if not. And you don't really have anything more than that. And even there, the level of confidence, it's often not very high, especially in these what you call infiltrated areas, right? Where there might be tumor, but it's very diffuse and there's not a lot of it. So first of all, you have to take out a bit of the tissue, which already is a danger in itself. It could cause bleeding. It could be taking out the part that is functional. And then you have to rely on information that comes back that is not very certain. What we want to do is instead to apply the needles to the bits of brain that is suspicious, and then uh, send it off for analysis, which is can be much quicker than that. And also, it can give you precise molecular information and tell you, yes, uh, there are some cells in here that have the precise characteristic of a tumor cells. You should take it out, or no, there are no cells of concerns in here.
So in effect, the major difference in that kind of use case would be you could sense whether tissue is healthy or malignant without removing it or damaging it,
And
and
with a lot more confidence.
with a lot more, even more accurately. And then is this kind of almost happening in real time? You mentioned the speed that, typically with a biopsy, you have to send it and, the labs figure it out and then kind of come back to you. Your kind of idea or your model is that there's a machine there in the surgery or just outside the room where you can just put the nano needles onto that and it comes up automatically.
Yes, that is the model. We're not at that model yet, but the model is, yes, having a machine right out of the operating room where you can steer it in and get a response. The machine at the moment is not something that we develop,
Right.
right? We use mass spectrometers or sequencing approaches, and the analytical side of the analysis still does take some time, which would probably still be in the 15 or 20 minutes time frame. But all of these techniques are speeding up by
Yeah.
themselves because there is a need to do it. So, realistically speaking, there is going to be a development in the next few years that are going to accelerate.
And 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 it's digital, which means it's much easier
Yes,
to accelerate than biological, right?
yes,
So
exactly.
effectively, what the needles are doing are they're converting the biological tumor or the biological material into something that can be into digital information,
Yeah,
which then you can do the analysis on
yeah,
super
yeah,
quickly
yeah,
and
yeah.
take advantage of what technology has to offer.
Yeah, absolutely. And obviously, I mean, again, this is not areas that we cover directly, but in terms of the diagnostic aspect, then you have these very rich, large data sets that you can analyze with machine learning and, artificial intelligence, and that can provide you very high confidence levels on what is happening.
Can I jump back a little bit to your example. I just had a I'm going a little bit backwards. We went forwards to brain tumors, but then I want to kind of jump backwards to the example where it's not a patient in surgery. They come in periodically, and it's in some respects what you're describing is a simpler way of kind of testing and something that's kind of less invasive. Is there a model where the patient doesn't have to come in, like it's almost like self-administered? I mean, I could imagine that if it's something on, say, the outside, like on your skin, you could take it out and
Well, I mean, but even if it is in your mouth, you can definitely, do it yourself. I think that we're not yet at the point where we have thought about these, how this can be deployed in that type of settings. And I don't think that there are any scientific challenges, but there are clearly regulatory challenges in delivering it in that type of setting. But there is, I mean, obviously, it's an extremely appealing approach, and it fits within sort of the distributed healthcare model that is going to be extremely valuable in the future.
yeah.
Uh, I think there would be a lot of sort of ergonomics and human design work to do to understand how a patient would self-administer that, and how reliable that information would become. But again, that is outside of my realm of expertise.
Yeah. As we have this conversation, I'm learning a lot from it. So so first of all, thank you for that. But the I think what you're presenting is you're at the cusp of three major innovation trends that are happening in healthcare today. One is about health at home, outside of the kind of the clinical environment. Um, Another one is around disease prevention.
Yeah.
So we have a system of sick care versus healthcare,
Yeah.
and so prevention is a huge um, trend for health innovation. And then the third one is personalized medicine and precision medicine, and it feels like what you're enabling is all three of those things.
I would say that yes, at different degrees and in different ways, we do enable values aspects of these key trends, and just by changing tack a little bit, when you talk about healthcare versus sick care, coming from a completely different healthcare system, growing up in Italy, uh, and maybe a sort of a different cultural background, I feel that. There has been a lot more healthcare being developed in Italy from earlier on, and it does reflect in a much prolonged, not only lifespan, but also quality of life in, when you become elderly. And uh, also, the burden on the healthcare system is significantly lower if you can do prevention at the population level rather than waiting until late and having to do treatment. So I think uh, that is gonna be an extremely challenging sort of s- transition for the NHS and for the way in which the NHS is set up, and that, for example, GP practices will have to change significantly the way in which they operate to make this happen, but it would be extremely beneficial if it can be pulled out.
This isn't really the focus of the the conversation today, but I'd love to just dig into that just for a second. Like, what I mean, it's one of the kind of the fortunate things of someone like you having lived in many different countries. You lived in Italy, or you grew up in Italy, you lived in the US, you're now living in the UK. So you have these different reference models. A little bit the same for me. I grew up in the UK, I lived in the US, I lived in Asia for a really long time, and then back here, and you kind of see that um, the way things operate don't have to be that way. But then also there are things that I really appreciate about the system that we have in the UK, that
Absolutely.
if I hadn't seen health elsewhere, I wouldn't necessarily have that appreciation either. But just on that point of prevention, like what would you say are the kind of the major characteristic differences between how it's approached in Italy and how it's approached in the UK?
I think that the biggest difference is cultural, as always, and it really comes down to the individual and the population. So in Italy, people do seek medical care very early for every symptom, and from at really early stages, and for things that maybe are not really concerns. And there's a lot of healthcare that is delivered through pharmacies, and a lot of signposting that is delivered through pharmacies, and as well as GPs practices, and that helps a very early sifting and triaging of things, as well as there's a very high awareness within the community of going to seek for healthcare very early. And yeah, but
So
and because it's in a way a cultural shift, it's a bit difficult to put your finger onto.
Yeah, it is, but I think what you've described is a systemic shift, actually, right? It's take it like for us, the first in the UK, the first point of care is your GP. So that puts enormous pressure on GPs, and then also as an individual in that in this system, it's hard to get GP appointments. So there has to be something really wrong with you for
Yeah.
you to actually turn up at a GP, because the friction is so high there. So the systemic uh, difference that you just kind of painted is one where there's more um, community health options delivered through the pharmacy, which then kind of alleviates a little bit of the pressure, I think, from GPs. So I think that's really interesting. I also think technology can do that as well, and so maybe we have the opportunity to skip a step and use technology almost as our first point of call for this.
Yeah, no, absolutely. I mean, obviously, I mean, the model that I was discussing is the model that exists and had developed before
Yeah.
the technology options that we have now. But you can already see the differences that even the simple wearable technology that is widespread nowadays is making in preventative care. I mean, the example of the Apple Watches and how their current ability to look at irregular heartbeat has actually improved the early detection of cardiac issues is significant. But it needs to be available, for these things to work. They need to be available and deployed very capillarly within the community, and it's a big investment.
Yeah. It's the investment, but then also it goes back to a little bit what you were saying earlier around deployment, because you speak to a lot of, say, cardiologists, and the devices and wearables is actually a massive frustration for them, because they're like, well, we you're rolling up with your Apple Watch data, and I don't, one, there's a lot of different devices. Two, I don't know how you've used that device, and and three, it's a lot of these things aren't really controlled. So the deployment is the workflow piece is a really big piece of that, right? I feel like this is a really good time to talk about the future, and this is a topic that I don't know if this will be true with you or not, but it's a topic I sometimes have challenges with, especially with scientists and researchers, because um, often there's a little bit of a hesitancy to say things that might not be like like that haven't been built yet, and may be true, may not be true. So I'm gonna kind of ask us maybe to use our little our imaginations a little bit, and also be, I suppose, hopeful. Um, with what you're building with nanotechnologies, what do you see, say, the future could be like? If everything goes super well, you talked about 5 years. the deployment for your technology in a clinical setting. But maybe if we fast forwarded to, say, 15 years, so we're now 2040, like, what do you think could be possible?
I mean, I think I'd rather, as you talked about hesitancy, I'd rather talk about the trends that are there,
Yeah.
where we could, see important things happening, and they will happen whether out of our laboratory or whether out of some of our colleagues that are working in this kind of space, because there's clearly a push and an interest in getting there. And obviously, one which everyone knows is advanced therapies, right? So gene therapies and cell therapies are going to become uh, widespread available in 15 years or maybe 20. We see them all already being deployed. CAR T cells therapies are in the clinic. They're making a big difference. Uh, there hasn't been such a big step in cancer therapy since the introduction of chemotherapy agents than what the cell-based immunotherapy are providing us. So there will definitely be more of that. And nanotechnology is a fundamental enabler of these type of therapies, because they allow you to do cell manufacturing in a way that is less expensive, in a way that is more robust, and that is safer than the tools that we use now, which are viruses, right? Because at the moment, for transfecting and for generating these uh, edited cells that you need, you just use viruses, which work fine, but have potential safety concerns, as well as immunogenicity concerns, and other aspects. So I think that cell and gene therapy is something where nanotech is going to provide a big support. The other one is the big transformation that we have already seen. I mean, lipid nanoparticles have been trying to break crack the issue of delivery for cancer for decades, and they've gone nowhere. And then COVID came along, and they were essential in developing the successful vaccine, especially the Moderna one, which is delivered with lipid nanoparticles. That was a transformational moment for all of nanotech. The 20, 30 years that we had invested in developing and understanding how these things work, all of a sudden, they really made a huge impact for the whole world. I mean, it's the thing that allowed people to go back and go back to work, go back to meet their friends, and do all this. So we will definitely see more of nanoparticles in vaccines, including cancer vaccines, for example, which is a new frontier. And the third aspect is this aspect of precision medicine, molecular diagnostics, and things like that. Again, COVID has been a big enabler there. The lateral flow device, uh, have, they are underpinned by nanotechnology. The little stripes that you see in red, or the cheaper ones in gray, are gold or silver nanoparticles. That's why I was telling you that gold nanoparticles are red. That red stripe that you see on there is a gold nanoparticle, and it only works because the nanoparticles can move through the lateral flow device. Um, there's a lot more coming in terms of diagnostics at the molecular level that is enabled by nanotechnology, especially in what is now the big frontier, which is what is called spatial omics. So being able to get genomic, transcriptomic, proteomic, and now even metabolomic information, but not like we did until now in a soup, so all your cells together, but your cells in the specific place. And hopefully, this is gonna be a breakthrough for the very complex diseases of our age. Things like dementia, things like ALS, all of the neurodegenerative diseases, uh, and many others, where it's not one single type of cell that causes the problem. It's the interaction between different cell types that is the problem. And by looking at how this happens in space, we're going to be able to understand better how this works. Where we think we can contribute in here is that we're now very good at looking at how, or there are commercial technologies that look at how the molecular information occurs in space, but nobody can look at it in space and time together. And all of biological processes are dynamic. They are inherently dynamic. They change in both space and time. With our nanoneedles, we can sample the same thing multiple times, okay? So we can, and then we have a spatial map. So we can get the space and time information together. And that's where we're heading.
CAR T is really powerful, and I just wondered if you would just explain like what that is kind of in kind of simple terms.
Yes. So in simple terms, you in your body, you already have your immune system, which is extremely effective at getting rid of any unwanted things, including very small tumors. So there's a lot of cells that start behaving like tumor cells that are taken away and killed by your immune systems before they develop into a tumor. And that is extremely effective. The problem is that when tumors grow, they then become able to sort of tell these cells that no, there's no problem here. And your immune system believes that, okay? That's one of the big, they call it hallmarks of cancer, okay? Being able to sort of evade the immune police. What you do with CAR T is you introduce into your T cells, into your immune system cells, a molecule that can recognize the tumor again. And then it tells to the cells to ignore everything else and go there to the tumor and go there and do its job, which is extremely effective, and it is that of killing the tumor cells.
And it's the nanoneedles that will deliver that.
Yes, that's the thing.
And the second question I want to ask you, and I didn't stop you here, you said genomics, something omics, something else omics, and something else omics. Can you, I think there were 4 of them. Can you just give me the 4 of them and then a very simple explanation of what they are?
There's a lot more omics than those, but
Oh my God. I'm not ready. I'm not ready.
Everything. So omics is a very, very general terms to mean all of the, right? So the genomics is all of the genes, and it's the first one that was developed, right? So it's basically, it allows you read the code of all of your DNA, and you know exactly all of the genes that are present in that specific individual, because as we said before, some people have genetic mutations, right? And all of us have what are called genetic variants. So that's why everyone is different from every other. But that is only the basic level, right? Then you move to the transcriptomics. So in biology, what happens is that you have your DNA. Your DNA gets copied into RNA, and then your RNA gets transcribed, so gets transformed into amino acids, which then become a protein. Okay, and then proteins do everything. Proteins are the effector, are what makes biology work. But the code is in the DNA, and it gets converted into the RNA. So genomics means decoding the DNA. Transcriptomics means decoding all of your RNA, because not all of your genes are expressed. Not all of your genes become RNA, not at the same level, not in the same way. And then proteomics is knowing from your RNA what you make into proteins. And then metabolomics, even further, is your cells, then your proteins do something. And by doing something, they use up some sugars, and they make them into other molecules. They change the lipids, and they do a lot of things. All of what your proteins work on are metabolites. And the metabolomics is exploring all of this. And because the balance of these is necessary for cell function as well.
What a fantastic explanation. You might have a career in like teaching sometime if you want one. You're already doing it. I've got one last question for you, and you don't have to answer it if you don't want. And it kind of comes from science fiction, because when I hear of nanotechnology, in my head is Star Trek nano robots floating around my body and curing disease. Is that ever going to happen, or is that always firmly in the world of sci-fi?
So nanorobots already exist in the sense, and they're not being deployed in the body yet, but they can move and they can perform actions both when they're stimulated or when they respond to how their environment look like. So nanobots are there. They just don't have the functionality or the function that we want them to have, nor they've been tested in the very challenging environment that is, inside your blood vessels or something like that. But they're coming. Their first use is not going to be what they're doing with Star Trek, but they will eventually happen.
Incredible. All right, that leaves me with hope. Ciro, this has been a really incredible conversation. Thank you so much for joining us today.
And thank you very much for having me, Dom.
Thank you.
Thank you.